Hello everyone, welcome to India's largest online learning platform ITLS Academy. Our today's lecture is about the human embryonic stem cell, and we will also see the human embryonic germ cell. This is part 2 of this particular topic. We have already covered many things regarding this topic in our previous video. So let us start with human embryonic stem cell and the human embryonic germ cell. Firstly, we will see pluripotency of human embryonic stem cells and embryonic germ cells. As we have discussed earlier that pluripotency is nature of any cell by which that particular cell can give rise to a lot of number of cells. So now we will see pluripotency in case of human embryonic stem cell and germ cell. Very first point is human embryonic stem cell and embryonic germ cells in vitro, that is outside the body, are capable of long-term self-renewal. Self-renewal is a property by which any cell can renew itself, or we can say if this is a particular stem cell, then it can give rise to same type of stem cell, same shape, size and everything, each and everything is same. So this will be known as self-renewal, while retaining a normal karyotype. Karyotype will not be changed. As stated earlier, a truly pluripotent stem cell is a cell that is capable of self-renewal and of differentiating into most all of the cells of the body, including cells of all three germ layers. Three germ layers are ectoderm, endoderm and mesoderm. These are three germ layers. Ectoderm is outer covering, endoderm it is found inside the ectoderm, while mesoderm is middle portion of any particular part. So our next point is cultures derived from embryoid bodies generated by human embryonic germ cells have less capacity for proliferation. Most of these cells will proliferate for 40 population doublings, that is, the maximum reported is 70 to 80 population doublings. Here in this diagram you can see this is blastocyst. We have discussed earlier in detail what is blastocyst and how it is formed. For reference, I am telling you that this blastocyst contains an outer layer that is known as trophoblast, while it contains a cell of mass, a mass of cell located at an end. This is known as inner cell mass, or in short it is ICM. This structure is blastocyst. The isolation of this blastocyst, this is culture. culture of this blastocyst, it forms embryonic stem cell colony. After colony formation, there is differentiation, that is pluripotent differentiation. This type of differentiation means it can form a number of cells, a lot of types of cells. Here, this forms the three germ layers. Three germ layers are ectoderm, mesoderm, and endoderm. If we talk about all these three layers, so this outer covering will be ectoderm. This middle portion will be mesoderm, or I can draw a line inside it. This line will form mesoderm, and in between the ectoderm and mesoderm, a third layer is found, that is endoderm. The only test of the in vivo pluripotency of human embryonic stem cells is to inject them into immunodeficient mice where they generate differentiated cells. Differentiated cells means their shape, size and work is changed or differentiated, that are derived from all three germ layers. Next point is two aspects of in vivo pluripotency. In vivo means inside the body. So two aspects of in vivo pluripotency typically used in animals have not been met by human embryonic stem cells. Evidence that cells have the capacity to be injected into a human embryo and form an organism made up of cells from two genetic lineages, and evidences that they have the ability to generate germ cells, the precursors to eggs and sperm in a developing organism. These two aspects are theoretically considerations. These are theoretical, however, because such tests using human embryonic stem cells have not been conducted. Comparison between human embryonic stem cell and embryonic germ cell. Very first point is, in both cases, the cells replicate for an extended period of time, show no chromosomal abnormalities, generate both, that is XX genotype is of females and XY, that is male genotype, and here generate both cultures, that is male and female cultures. And express a set of markers regarded as characteristics of pluripotent cells. However, the embryonic stem cells derived from human blastocyst and embryonic germ cells differ not only in the tissue sources from which they are derived. They also vary with respect to their growth characteristics in vitro and their behavior in vivo. In vitro means something that is outside the body, while in vivo means something that is inside the body. In addition, human embryonic stem cells have been propagated for approximately 2 years in vitro. For several hundred population doublings, whereas human embryonic body derived cells from cultures of embryonic germ cells have been maintained for only 70 to 80 population doublings. Several research groups are trying to grow human embryonic stem cells without feeder layers of mouse embryo fibroblast, in short known as MEF. And this is very labor intensive to generate. Here you can see difference between embryonic stem cell and adult stem cell. If we talk about embryonic stem cell, so these can become all cells of the body. Embryonic stem cells have the ability to become each and every cell of the body. Pluripotent and totipotent differentiating capacity. They have differentiation capacity. While in case of adult stem cells, these are generally limited to differentiating into different cell types of tissue of origin. Mostly multipotent differentiating capacity. Next point in case of embryonic stem cells is large numbers can be easily grown in culture. These are difficult to control proliferation. Proliferation is the ability of the cells to divide, or we can say to replicate, or to generate their progeny. While in the case of adult stem cells, these are rare in mature tissues and methods of expanding in cell cultures has not yet been worked out. Next point in embryonic stem cells is that these can cause transplant rejection. Transplant rejection is a process in which any organ that is transferred from the one body to another body, so the body to which that particular organ is transferred, that can reject that particular transplant or organ. The organ that is transferred is known as transplant. So we can say that embryonic stem cells are responsible for the rejection of graft or rejection of transplant. While in the case of adult stem cells, patient's own cells can be expanded in culture and reintroduced into the body, thus causing no rejection because that particular patient has their own cells. These cells are not transferred from any other body, or these are not taken from any other tissue. These are their own cells. That's why there is no any kind of rejection of graft. Further, if we talk about embryonic stem cells, so these are virtually immortal. This is due to high expression of telomerase enzyme, which is important for maintaining chromosomal stability. Telomerase is an enzyme that is mainly used in the process of replication to protect the end of DNA or RNA. To protect the end of DNA, this telomerase enzyme works on the ends. So if we talk about adult stem cell, so in the case of adult stem cells, these undergo senescence. Senescence is the process of decay steadily, or you can say if this is particular, this is initiation, this is growth, and this decline is known as senescence. So these adult stem cells undergo senescence. So we can say that these cells decay and considered to have a finite lifespan, that is, they have limited lifespan. Telomerase enzymes absent in most mesenchymal stem cells. Most of the time, telomerase enzymes are absent from here. Potential uses of human embryonic stem cells. The most often discussed is their potential use in transplant therapy, that is, to replace or restore tissues that has been damaged by disease or injury. However, treatments for any of these diseases require that human embryonic stem cells be directed to differentiate into specific cell types prior to transplant. So now we are going to see using human embryonic stem cells for therapeutic transplant. Using human embryonic stem cells for therapeutic transplants failure and osteogenesis imperfecta. Next point is one of the current advantages of using embryonic stem cells are as compared to adult stem cells is that embryonic stem cells have an unlimited ability to proliferate in vitro, that is inside or these have the ability to proliferate outside the body, that is in vitro, and are more likely to be able to generate a broad range of cell types through directed differentiation. The potential disadvantages of the use of human embryonic stem cells for transplant therapy include the propensity of undifferentiated embryonic stem cells to induce the formation of tumors, which are typically begin because it is the undifferentiated cells rather than their differentiated progeny that have been shown to induce teratomas, that is, tumors. Tumor formation might be avoided by devising methods for removing any undifferentiated embryonic stem cells prior to transplant. Here in this diagram you can see this is human embryonic stem cell. Human embryonic stem cell establish pure cultures of specific cell types. If we are going to test physiological functions in vitro, that is in outside the body, here we are going to see stimulated insulin release. For example, we are taking the example of insulin release. So, if we demonstrate efficacy for the efficacy in rodent models, in non-human primate model with the rhesus embryonic stem cell derived cells, that is diabetes and Parkinson's disease model in primates. Evaluate integration into host tissue, for example, cardiomyocytes for treatment of heart failure. Next will be recurrent autoimmunity. These all points to demonstrate efficacy, and after the testing of this efficacy, human trials are done. For the demonstration of safety, it will be firstly in non-human primate model with the rhesus embryonic cell derived tissues. Further we'll see show absence of tumor formation, so absence of transmission of infectious agents. If these points are done, then this will further go to human trials. If we are going to test methods to prevent rejection, so we will see firstly multi-drug immunosuppression. We will create differentiated cells isogenic to prospective recipient using nuclear reprogramming. Now we transduce embryonic stem cells to express recipient MHC gene and further establish hematopoietic chimera and immunologic tolerance. If these all points are successful, then they will again go for human trials. Here some points regarding establishment of pure cultures are given. You can check that lineage selection by cell survival or cell sorting and induce with supplemented growth factors. These points are regarding establishment of pure culture. Firstly, you have to select by the cell sorting, and then you have to provide growth factors for the culture. We are now going to see some other potential uses of human embryonic stem cells. So the first point is human embryonic stem cells could be used to study early events in human development. Still unexplained events in early human development can result in congenital birth defects, and placental abnormalities that lead to spontaneous abortion. Such cells could also be used to explore the effects of chromosomal abnormalities in early development. This might include the ability to monitor the development of early childhood tumors, many of which are embryonic in origin. Human embryonic stem cells could also be used to test candidate therapeutic drugs. Currently, before candidate drugs are tested in human volunteers, they are subjected to a barrage of preclinical tests. Although animal model testing is a mainstay of pharmaceutical research, it cannot always predict the effects that a candidate drug may have on human cells. For this reason, cultures of human cells are often employed in preclinical tests. So here is genetic manipulations of human embryonic stem cells. If we will talk about the MHC genes, so first we will see genetic manipulation of MHC genes. MHC is a major histocompatibility complex that play a major role in our defense system, or we can say in our immune system. So let us see genetic manipulation. This is embryonic stem cell. Here, if this is deletion of MHC gene, then it will result in MHC deficient tissues. If there is substitution of MHC genes, then this will result in self-MHC expression tissue. Let us see nuclear reprogramming. This is enucleated oocyte, that is, it does not have a nucleus. This is micropipette. We are going to do our experiment through this micropipette. Here we will transfer a nucleus that is donated from any other tissue or any other oocyte. This is holding pipette that will hold this oocyte at its place. Further it goes differentiation. Embryonic stem cells derived from nuclear transfer product. These kind of embryonic stem cells will be derived. Now it comes to tissue specific differentiation, and after differentiation, this differentiated tissue expressing donor nucleus MHC genes. Let us see hematopoietic chimera, complete, mixed and micro. These are embryonic stem cells. After differentiation, these will form hematopoietic differentiated. These are differentiated cells. Let us start with the starting. These are embryonic stem cell. After this differentiation, these cells will form further kind of cells. Here will be hematopoietic differentiation, and after hematopoietic differentiation, there will be co-transplantation. On the other hand, these cells are differentiated into muscles, pancreatic islet cells, and neurons. Further, it goes to co-transplantation. So, these are some examples of genetic manipulations. and these are of human embryonic stem cells. That how we can perform various things with human embryonic stem cells. So thank you guys for watching this session. Follow ITLS Academy on various social media platforms and also subscribe our channel ITLS Academy on YouTube. So thank you very much everyone for watching. If you like this video then please like it, share with your friends and also subscribe our channel ITLS Academy. Thank you very much.